So we've been talking about three main ways that microbes can acquire DNA from their environment or from another cell. What's one method? Transformation. Transformation is the simplest. It's taking up that naked DNA that's just laying around. And so that one, they are seeing some differences on how gram-positive bacteria do it versus gram-negative. So be up on that for the exam. Just in general, the gram-negatives are using that pili structure, whereas the gram-positives build that whole transformosome like tunnel. So transformation, what's another one?
Conjugation.
Conjugation. All right, that's with the plasmid transfer. Is it the whole plasmid that gets transferred? Yes, no, whole plasmid?
One of the two.
One strand, okay. I can't help but think, my mom used to tell me stories. She delayed me joining into kindergarten a whole year because she said I didn't like to share my toys. And she told me that much later in my life. I'm like, I still don't like to share my toys. But anyways, don't share your whole plasmid, right? That would be foolhardy. Don't do that. You can give one strand away, but keep back one strand so you can make a complementary copy and you're good. All right. So it's one thing to share, but don't give up the whole thing. And the reason I'm making such a big deal about this is you have to think about it from the microbe's perspective and through the lens of evolution. If that plasmid confers traits that makes that bacteria adapt even better to its environment, it would not give up its whole plasmid for a neighbor. Okay, so think about it that way. Conjugation. So that's the plasmid transfers through the sex pilus. Okay, so they have to make physical contact. That sex pilus is used, and they will get one strand transferred of the plasmid over. And usually the plasmid stays put, in the sense that what I mean by that is the plasmid stays circular around in the cytoplasm. But every once in a while, you can get a plasmid that pastes into the chromosome. And that's a special condition. And then if that happens, you can get, eventually that plasmid can come back out. And if it does that, sometimes it takes chromosome content with it. Okay, so conjugation is that plasmid idea. And then what's the other one? Starts with a T. The other method, transduction. So transduction has two versions. You have specialized and generalized. Okay, so we were looking at that transduction last time. Okay, so here's generalized transduction, at least part of it they're showing. And so that middle cell, that figure here in the middle with that bacterial cell, that virus is taking the cell hostage. It's immediately saying, make more particles, more viral particles. I don't really care about you, just make me a bunch of more viral particles. So you can see that the bacterial DNA is fragmenting, it's really chaos. And then the viral particles are getting packaged up in that lower image, and you can see there's different colors of DNA. So they're trying to show that sometimes everything goes perfect and you get viral DNA into the viral particle, and that's good. And then you see some cases where the host DNA got packaged in there. Now that cell isn't going to benefit from it. It will lyse open and all those viral particles will spew out, and then they're going to look for a neighbor microbe, a neighbor bacteria to do this all over again. But if a neighbor microbe gets the bacteria, the DNA from the first bacterial host injected in, that can be a good thing for that second bacteria, that neighbor, because that DNA from the first host can get pasted into the chromosome of the second host. And so, and usually what happens is that virus can't cause infection because it's faulty. It doesn't have enough of its own viral DNA to cause problems. And so that second host can actually benefit by getting a transfer of DNA from the first host bacteria into the second host, okay? Generalized, why do we say generalized? It's not a specific piece of the chromosome. So this is this gray blob here. It kind of does look like a plasmid, doesn't it? Because it's circular. But it's kind of interesting. Most bacterial DNA is circular, just really big circles. Plasmids are much smaller circles of DNA. And so here you see that circular, huge piece of chromosome getting fragmented, and it's just random what bits and pieces of that host DNA end up in the viral particle. So general, it's just general piece. So they should have said random transduction, but they chose the word generalized. Okay. And then we compare that to specialized. So that means there's a very specific piece of DNA that's selectively transduced. It's not just any old random piece of DNA that's floating around. And in order for this to happen, you have to get that virus injecting its DNA into the cell, and then the viral DNA gets pasted into the host DNA. When that occurs, we call it a prophage. And then they're showing that if the virus does its normal routine, it will cut out, eventually, it will cut out its DNA out of the bacterial DNA and take the cell hostage and say, make more viral particles. That's what's being shown on the left. So, but what we're focusing on is what's on the right. And you're seeing that now that viral DNA is getting pasted out, but not perfectly. I'm sorry, cut out, but not perfectly. You have some flanking bacterial DNA coming with it. All right, so there's a selected portion of DNA that's getting brought with it. It's the adjacent or flanking DNA, right where that viral DNA had been inserted. And then you get a mixture of viral DNA plus host DNA being packaged into the viral particles. When they say they're defective phage particles, that means that when they go to attack another cell, they won't be able to cause any problems. However, they will be able to give that DNA, that bacterial DNA, to the second host. So again, the second bacteria can actually gain from this. It won't get infected and go, and get lysed open because it's a defective phage. But that phage could be transferring some useful DNA from the first bacteria. Okay, so special bits of the host DNA where the phage DNA had pasted in there. And then this picture is just showing side-by-side comparison of general versus specialized transduction. And they use the word prophage in there. So when you say that term, please get this straight, because I've seen this in past classes that students don't understand the word prophage. So please get that set in your head by Friday. Prophage is a viral particle, a bacteriophage, right? And its DNA has been pasted into the chromosome. So let's see if I can find that better picture. Let me go to this one. When that viral DNA pastes into the bacterial DNA, it's still viral DNA. So they want to, make a point that, yeah, that stretches viral DNA, but it's not an active viral particle. So they call it a prophage. Okay? It's a probacteriophage and prophage. That means it's not quite a fully functioning, it's just the DNA sitting in there. Okay? So that is different from the word bacteriophage. It means two different things. Bacteriophage is the full viral particle. If someone says, oh, look at that bacteriophage, it's a virus that can infect bacteria. If someone says, oh, look at that prophage, it's just the viral DNA pasted in to the host chromosome. Two different, completely different meaning size. Make sure that you get those two terms straight. So here they're showing this idea that once you have a prophage scenario, they're actually showing that this can really benefit the bacteriophage, because once that gets cut out of the DNA, the host DNA, it can take the cell hostage. So that if you go over to the left here, that's what's being shown, that you can get the bacteriophage saying, hey, make more particles of me. And that's what's going on the left. So that's one thing that can happen. Another pretty sneaky thing is that the prophage just stays put in the chromosome. And as those cells are dividing and producing progeny cells, all of that prophage that goes with it. So without having to do much of anything, you get that phage passed into future generations. And that's a pretty lazy way for that prophage to get spread amongst the population there. So they think that most bacteria that hang out in the natural world and natural settings, they think most of them do have viral particles, viral stretches of DNA in their chromosome, just like we do. We have a lot of viral DNA in our chromosome too. And so they think that's pretty common. So they say most bacteria are lysogens. So when they say the word lysogen, they're referring to the bacterial host cell that contains viral DNA in its own DNA. Okay. So this is new information. So if you have a bacteria that has some viral DNA, and it's a prophage, and then you can see some interesting things that are important to us in the sense that it can make non-virulent, non-pathogenic bacteria become pathogenic. And so a classic example is Corynebacterium diphtheriae, which causes diphtheria, which is still a serious illness. It actually allows for this bacteria to produce what's called an exotoxin. So what happens is the prophage has a stretch of DNA that codes for this exotoxin protein. And it makes them pathogenic. So if you just have a normal Corynebacterium that hasn't been infected by a phage and hasn't been given that exotoxin gene, it's fine. Like I used to order, they call it avirulent C. diphtheria. I used to order that and work with it in class. But I decided to phase it out because I knew it was safe because there wasn't an exotoxin gene in there. But I found a better bacteria that actually gives better results for a certain test that I was using this one in. So I went with that instead. But anyways, Clostridium botulinum I have never used and never will. This one's a whole different story, even though it's the same idea. I just wouldn't want to touch it with a 10-foot pole. If it gets lysogenized, which means if a very particular phage puts its viral DNA into the host DNA, it can turn Clostridium into a very dangerous and deadly pathogen. It produces a neurotoxin in that case. And I have in the notes that the botulism toxin is the most potent toxin known. About 75 nanograms can kill an adult human. 75 nanograms. That's not much. 1 kilogram would kill the entire US population. The toxin is colorless, odorless, and tasteless. So you have no idea it's there. However, the good news is the toxin can be inactivated by only a few minutes of boiling. So if you've ever heard the advice that if you take like a can of soup out of your cupboard and it's bulging, like it's actually looks like the top is bulging, don't eat it. And if you absolutely must, like if you're in a scenario where you're starved to death, if you didn't have that, you should boil the heck out of that soup because chances are it's Clostridium botulinum in that soup. And the bulging is gas production. One of the things that it likes to do is produce gas as it's running its physiology. And it can produce so much gas that it can actually burst a soup can. Or, if you do home canning, that's the risk with home canning. You got to be careful that you're not introducing any pathogenic Clostridium. But if it didn't have that neurotoxin gene from the virus, if it was avirulent, it's fine. There's plenty of Clostridium botulinum that hangs out in the soil, and that's totally fine. It's not going to kill you. By the way, the reason that it does kill you is that it paralyzes your muscles. It's a neurotoxin. So it paralyzes your muscles in a relaxed condition, not tense condition. So it relaxes you so much that your diaphragm that helps you breathe, that relaxes too. So you can't breathe. So really, you die of asphyxiation. I can't say that word today. Because you can't take in any breath. Your diaphragm is too relaxed. That's why there's another syndrome that babies get from eating honey. People inadvertently give their babies honey, like 6-month-old or 8-month-old baby that has spores of Clostridium in it. And then the baby, they call it floppy baby syndrome. I don't know that they say that to the mother's face, but that's what it's called in the medical jargon. And that's when they bring in a baby who has had poisoning. That's what the baby's floppy. I mean, they're so relaxed, they can't hold any tone to their muscles. And so that's not as big of a deal as it used to be, but like back in my grandfather's era, that was a fairly common thing. And so now they say never feed children any honey until they're over 2 years old. Anyways, all right. So if you are a lysogen, if you're a bacteria hanging out in the real world and you have viral DNA pasted into your chromosome, you have those prophages in you, it makes you immune to the same exact phage trying to get in. Okay, so if the same exact phage particle comes along and tries to get in, and they don't quite understand why, but it just won't work. So it's almost like an immunity from that same phage. And of course, some of those genes that are on that prophage could be useful to you. Like it could make you pathogenic, and that could be useful from your perspective. It's not so good for the host, but if you can become a pathogen, that can make you it's an effective way to get access to nutrients. So it could provide a selective advantage to you. So one of the things about horizontal gene transfer is, we've mentioned it a little bit, is especially with conjugation, when it's a plasmid involved, so many plasmids contain genes for antibiotic resistance out there. I mean, you can buy them from companies. They sell these plasmids to do experimental research with, but they know that there's a whole bunch of plasmids out there that code for antibiotic resistance. And then if you think about your own body, you have millions upon millions of microbes hanging out in your gastrointestinal tract in close proximity to each other, and of course to you, because they're right inside your intestinal tract. And so you can get all kinds of interactions going on, which we'll talk about later this semester. But one of the concerns is if you end up in a situation where you get sick with an antibiotic resistant bacteria and it takes up residence in your GI tract, horizontal gene transfer could occur. So they're worried about, how likely is that? What can we do about this? And so it's something that medical microbiologists certainly is thinking about, and they're trying to figure out ways to stop microbes from doing this, sharing horizontal gene transfer of especially antibiotic resistance. So it's bad enough that we have microbes out in the real world that are antibiotic resistant, but then if they get in you and start sharing that with other microbes, then, that's something definitely to be worried about. And that is an active area of research. They are looking at this pretty carefully because that's kind of a nightmare scenario. So here's just a visual trying to show in the gastrointestinal tract, you have, of course, the lining of your intestinal tract is filled with epithelial cells. Microbes can hang out in really close proximity to that, and of course, they're hanging out with each other. And so they're trying to show that you can have all versions of the typical horizontal gene transfer going on amongst all those bacteria, because they hang out so tightly together. They're very intimate, very close association physically. So we'll talk a little bit more about this when we get into microbe-human relationships. And we'll also talk about how microbes can also directly influence you and your cells in your body, not just your intestinal cells, but your whole body, all the way up into your brain. So we'll talk a lot more about that in a little bit later. Then don't forget, microbes are good at getting rid of genes that no longer serve to give them any kind of advantage to their habitat. So they can do that quite readily. So genome reduction is definitely a thing with microbes. They're really quite savvy at doing this. They've studied the Mycobacterium leprae genome, and it has a bunch of what they call pseudo genes that are no longer functional. And they think that's, it's going through the evolutionary process of purging things that it no longer needs. And it's probably because, Mycobacterium is an obligate pathogen. And so it's kind of living the sweet life. And why should it carry all these other genes that it needed when it was free living? Now that it has a host to sponge off of, it really doesn't need a lot of those genes anymore. And the kind of the flip side is this idea of gene duplication. Remember, microbes are haploid, okay? However, they can generate new genes through this idea of gene duplication. And I like to think of it as a microbe. It usually happens during cell reproduction when they're copying their DNA to before they split in two. The duplication event happens usually during that time. And I always kind of think of it as it's an extra gene now that they kind of play around with, depending on their habitat. They could turn it into something useful. And so it can free a gene from its previous functional constraints and allows divergent evolution through mutation. And so I kind of imagine them playing around with it, like, is this going to help me if I do this or that or this? Of course, this is all through selective natural selection and adaptation. Okay, so genome reduction and gene duplication. And then this is just a fun thing that they had in your book, looking at this idea of DNA that's coming from other sources that are found. And so they're saying this transfer of DNA, this horizontal gene transfer idea, they're saying over the course of evolution may be more common than previously realized. And so this is what Dr. Lynn Margulis was saying with the endosymbiosis idea. And so a lot of her other research was pointing to this, that you have this symbiosis going on through evolutionary time, leading to the idea that you can get genes showing up in other organisms that were from the original bacteria. And so you see this idea of, and we'll talk a little bit more about that when I said that, viral DNA is in the human chromosome, and it has actually given us some adaptive and definite advantages as we evolved on Earth. Okay. All right, so that's the end of horizontal gene transfer. So transformation, conjugation, and transduction are like the three typical ways we talk about horizontal gene transfer. But in a little bit, I'll also talk about a couple of other strategies that have been recently discovered that now they're saying, ooh, this would be another great way for gene transfer to occur. And so we'll get into that when we get into cell structure and function a little bit more down the line here. So speaking of cell structure and function, let me just introduce that topic before we talk about the exam. Okay. So this is the information on cell structure and function. So most of what you're gonna see under the microscope in lab will be bacteria that are coccus shaped or rod shaped. And we do want that terminology. By the way, the old way, you might see it in some readings or on microscope slides and they're labeled a certain way. The old way of saying rod shaped bacteria, rod shaped, the old way of describing that, we would say bacillus. Actually, I'll put it in the lowercase. We would say it's bacillus shaped. However, I think you've already worked with the Bacillus genus in lab, right? You looked at, can't remember if it's Bacillus megaterium. So that's actually a genus name, Bacillus megaterium, for example. That's a species. And so it got really confusing. Someone would say, oh, look at Bacillus. And you're like, what? Which one? Bacillus megaterium, Bacillus cereus, Bacillus subtilis? And they're like, no, I'm saying it's rod shaped. So they said, let's just stop using that. That's confusing. So they went to rod. All right. But you'll see in the old literature, it'll be called a bacillus. So try not to get confused by it. But there are other shapes they're trying to show. Spiral shaped. That one is fairly frequently found out in the real world. Spirochete, we really don't want to see any of that in our lab. Those are usually pathogens. So we don't want those. Although, I mean, there are some non-pathogens, but the ones you would see in a lab are usually ones that have been dangerous. Then you get these weird shaped bacteria that look pretty different, where you can tell there's like two different parts to them. And so a lot of bacteria, for example, that hang out in rivers and streams, they tend to be, a lot of them can be stalked or have this protrusion like that, and they often use it for attachment. So if there's, running water, and they want to stay attached to a rock in that river, they will often be stalk shaped. And then they'll kind of, dangle in the stream current as the nutrients flow by them and they absorb those nutrients. And then filamentous bacteria are fairly common too. The Streptomyces bacteria that I work with are filamentous, and you can find, we once did the first day of microscope use in this class, the idea was to scrape your mouth, like your gum line, and look at it under the microscope. And I remember one student had a bunch of filamentous bacteria under the slide. He was freaking out. I'm like, no, it's okay. That you actually, I would guess most of us in this room have filamentous bacteria in our mouth, and they're totally fine. So don't worry about that. But it is kind of different looking. So, okay. So microbes have a cell membrane and a cell wall, right? The gram stain, we were looking at that idea on Tuesday in lab. But underneath that cell wall is a cell membrane. And so it's the same basic idea as what we see with our membranes. It's a phospholipid bilayer, which is what they're showing there in figure B. Figure A is just showing the detail of what the phospholipid looks like in chemical structure. Okay, so you have the hydrophilic regions on the outside and the center portion is hydrophobic. That's where you see those fatty acids dangling there to form that bilayer configuration. Then, of course, microbes have a lot of proteins in their cell membrane. It can go all the way through, so it's transmembrane if it goes all the way through, or maybe it's just on the inner portion or the outer portion. So the cell membrane of a bacteria is cytoplasmic membrane or cell membrane. It's about 6 to 8 nanometers wide. It has lots of proteins, and they say it has a consistency that's somewhat fluid. You want it to be somewhat fluid. That's a good thing. Now, microbes don't have true organelles, although we'll talk about that. That's kind of debatable actually. But right now, what they're saying is, let's just say they don't have true organelles. And so they don't have mitochondria, for example, like we do, where we get a bunch of ATP made. So where do they do that? They still have to get their ATP, but they don't have a mitochondria. So what they do instead is they use part of their cell membrane for that. All right, so a portion of their cell membrane will be devoted to running an electron transport chain to get ATP. We do it in our mitochondria, but since they don't have that, they'll do it in their cell membrane. So for a microbe, their cell membrane is super important. Arguably more important than our cell membranes are to us. Because we have all those internal organelles that specialize in doing stuff for us. We don't have to put everything in the cell membrane. Microbes have to do, so much of what they do physiologically takes place in the cell membrane. And so it's really important that they have a nice, healthy cell membrane. So here's the ATP synthase, you can see embedded in the cell membrane. And so, they're getting their ATP by using the cell membrane and running their electron transport chain process. We do all that in our mitochondria. They can have sterols in their membrane that helps strengthen the cell membrane, gives it some strength. So you see that in fungal cell membranes. Hopanoids are the same idea as a sterol, but they're chemically slightly different. So you can't exactly call them a sterol, so they named them hopanoids, but it's the same idea. They're lipids that are add a little oomph to the cell membrane, a little bit of strengthening. Okay, and you see that in the bacteria, domain bacteria. So we have sterols in our membrane. We're eukaryotic. Fungi are eukaryotic. They have sterols, but bacteria tend to have hopanoids. And this is just the chemical comparison. You don't need to know the chemical structure, but you can see on the left, that's a sterol. The middle one is cholesterol, which you've heard quite a bit about, I'm sure, over time here. And then the hopanoids, that structure there. So you can see that they're related. And here they are in context to the cell membrane. So you can see that they just add a little bit of structure and rigidity, but you still want the membrane, somewhat fluid. So they can actually test for the presence of hopanoids, like in a soil sample, if you want to look at, microbial populations, that's something that they can actually do. They'll actually run a test for hopanoids because it's so distinct to bacteria. Okay, so then in the cell membrane, when I showed you this picture, well, let's go to this one. So these are fatty acid tails, right? So, okay, that's what we have. We have fatty acid tails. Bacteria have fatty acid tails. So that's what's being shown here in figure A. The R group is your fatty acid. Now, what they're showing here is that where you have the glycerol hooking into the fatty acid, you have an ester bond. If you look at domain archaea, they don't have an ester bond, they have an ether bond. Organic chemistry. Having nightmares yet? I am. So ester versus ether. I know that sounds like, well, what big deal? It is a big deal because you can actually use this to identify who's in a sample just by looking at ester bonds versus ether bonds. So it is a big deal. And I need you to know that, okay? So in the domain bacteria and in us, domain eukarya, you have an ester bond connected to the fatty acid. In domain archaea, it's an ether. And then the other weird thing, this R, instead of being a fatty acid in the domain archaea, it is an isoprene unit. Okay, so down here I say in figure B or domain archaea, the R group is isoprene. So this is figure C here, that's an isoprene unit. So you'll have multiples of these stuck together. And that's the tail. It's an isoprene tail, not a fatty acid tail. So the cell membrane of archaea are quite different compared to the domain bacteria or the domain eukarya. So let's look at that a little bit. So here with the archaea, all of these squiggles here in the blue, that's all isoprene units, okay? Now, what they're showing is a couple of variations, depending on the archaea they're studying. They can have some pretty dramatic differences. So. Yeah, that's in the next one. So this top image and then, so figure A and B, what they're trying to show is sometimes you'll have these the ether link to a bunch of isoprene units. And then you'll have the gap, and then you'll have the same thing on the other side to form a bilayer. Okay, so let me go to this picture. So here. So this is your bilayer, okay, but these are not fatty acids, these are the isoprene units, they call them phytanol. Don't need to know that, I'm more concerned that you understand these are isoprene units. But it still forms a bilayer. So it's similar to the phospholipid bilayer that we have with our fatty acids in us. This one, they're saying, sometimes though, archaea have been discovered that don't have a bilayer membrane, it's solid. It's just solid all the way through. So they say it's a monolayer, not a bilayer. And so they're really weird. So they're trying to show that here, but there's no break here. Where there's to create that bilayer configuration, it's just solid isoprene units and they're hooked all the way solid. Then they call that a monolayer. So they've discovered many archaea that are like this. Typically, when they discover these archaea, they're from habitats that are very hostile to us, like hydrothermal vents or deep in the ocean floor, you know, just in the sediment where it's really cold, or in volcanoes. So you can see that it's usually a really hostile environments to a human. That's where you often find monolayers. So they think it helps like extreme heat, and especially with extreme heat, it helps keep the cell membrane together. It gives enough structure that they won't like melt apart at extreme heat. So think of it that way. Questions on that? And then this lower image is just trying to show sometimes they put these interesting little structures in there as part of their design. So the cell membrane of the domain archaea have some interesting options that they can do. And it's really has to do with what habitat are they living in. That really influences how they build their cell membrane. Sometimes they can get away with just a bilayer membrane like what we do, but sometimes they have to really do some interesting adaptations in order to survive. Does that make sense? Okay. So again, just trying to get across the point that the cell membrane is very, very important for the microbe. They have to do extra stuff in that cell membrane that we don't. So they have extra proteins and enzymes embedded in the membrane. And that is how they also generate a proton gradient across the membrane. They use their cell membrane. They don't have any other membranes really, the typical microbe anyways. They have to use their cell membrane in order to do a proton gradient across a membrane to get their ATP, right? Okay, we're gonna stop there. And let's switch over to thinking about the exam. Let's do that. I still have not finished making it, but I think I told you last time there's about 28 multiple choice questions. Still stands at that, and 2 essay question. No, very good question. Thank you for asking. I will not ask that information. So it's through gene transfer. So essentially it's 3 PowerPoint slide sets and the associated readings. So it's not a ton, but please study because this is low stakes. There's less for you to focus on. Typically what I see is students do really well on this first exam. And of course that obviously helps you as you move forward through the semester. It puts you in a more comfortable position for, starting out strong. Okay, whoops. Let me do this. All right, so the first slide set. Okay, so thinking about microbial evolution and really the idea of cellular life. Yes.
Hopefully like issue wasn't on the lecture slide, based on, or is it mainly based on the slides?
Good question. So there will be some questions coming from the textbook readings. Okay. That I haven't talked about. So make sure you pull up those ranges on the Word document of which page ranges I want you to know. So there'll probably be a question, one or two questions about evolution of microbes and like how did cellular life arise on planet Earth? And that's covered in the textbook, but I didn't get into it in huge detail in class. However, having said that, the vast majority of points when I make exams come from the PowerPoint lecture slides. Okay, and then a much smaller percentage from textbook readings that I didn't talk about. Much of what I assign you to read in the textbook is stuff I talked about. It's just presented in a slightly different way because I didn't write the textbook. Okay, so it's another voice for you to use in case what I said didn't make sense to your brain. Go to the textbook, see if that helps, is always a good strategy. Okay, so in any class, when I'm making an exam, the first thing I do is pull up my PowerPoint, sit there and go, okay, I got to ask some questions. And so I'm literally staring at the PowerPoints going, okay, how can I make this into a multiple choice? How can I turn this into a drag and drop match or whatever? Okay. So definitely the time we spend together in lecture and those PowerPoint slides, your notes, that's critical. I know some professors kind of flip it and do more off the textbook, and that to me, that doesn't make sense. We're spending all this precious time together. This is what's important. So, okay. Questions on slide set 1 on microbial evolution? Got into a few terms like stromatolites and what those are. And then we started talking about just kind of the general idea of the formation of cellular life, protocells. Stromatolites in the UP. I often don't ask you to do this, but for this, I will ask you to know like Carl Woese's name, Lynn Margulis's name, and then we talked about Richard Lenski's evolutionary experiment down at MSU. So I will be asking those names because they're like huge in their fields of study. Very, very famous. They did huge contributions, especially Woese and Margulis. Lenski's still alive and doing wonderful things, but the other two have passed. Questions on anything here? You comfortable with the endosymbiosis theory? What was the outcome of the endosymbiosis theory when you think about it? What did that allow to happen? So you have a bunch of protocells all hanging out together, swimming around. You got the whole endosymbiosis process going. But what was the actual outcome? Yes. So what kind of cell type have mitochondria and organelles? Eukaryotic. So I've asked this question before, and it's surprising to me how many students just don't know the answer. I mean, when you think about they can describe the endosymbiosis theory, but then which is great. I mean, be able to do that, the basic idea of it. But really, the outcome was it created a brand new cell type that had never existed before. You went from prokaryotic cell type, where all the protocells were hanging out together, and then through the endosymbiosis process, you end up with a eukaryotic cell type. That's a phenomenal shift in the evolutionary process. It's huge. And so let me see if I can find, is that in here? Let me go to this. So this phylogenetic tree. So here they're actually showing, they're trying to show it. Yeah, the mitochondria were once free living. They were once free living bacteria that figured out how to use oxygen when most other bacteria were killed by oxygen. And then here you see the chloroplast. Again, trying to point out, yeah, these were once free living. The chloroplast was a cyanobacteria, right? So what they're trying to show is that the chloroplast evolved from a cyanobacteria getting engulfed, and the mitochondria was once a free living proteobacteria. So that's how you interpret that. You see this line going off to the proteobacteria and then the mitochondria coming off that line. So what they're saying is, yeah, mitochondria was once a free living proteobacteria. Chloroplast was once a free living cyanobacteria. It just went endo. It became an endosymbiont. All right. Any other questions on the first slide set? Woo. Where'd it go? No? Golden? Second slide set? Let me just go. A little bit about, how do we study the evolutionary process and speciation. Some of the methods, we touched on a couple different methods. Questions on any of that? Yes.
Analysis that we talked about in class.
Yeah.
It just wants to be like familiar or be like very in depth with what we know about.
No, so know that, you have the one that I wanted, the number I wanted you to know more than any other was where did it go? This one. 95% greater in the 16S ribosomal RNA. That 95% I want you to know. The other slide we were just looking at, the genotypic analysis slide, the general idea, but you don't have to know all the numbers. Other questions on this? How do the idea of how microbes have their version of sex, that definition, I would like you to know. The one that Dr. Margulis and Dorian, her son Dorian Sagan, came up with. Since they don't have sperm and egg, we got to think about it a little differently. And then don't forget to think a little bit about Dr. Lenski's experiment. That was a big deal. Going from how we define E. coli, we say E. coli should not be able to use citrate. That's part of its definition. And now he was able to, through this process, get it to use citrate. Oh, we're running late. And then the gene duplication, the gene transfer slide, we just went over earlier in this. And I did ask you quite a bit of questions as we're going through it. What are the three methods? What's the difference between special and generalized? And so, yeah, that information definitely be up on that for the quiz, the exam. Okay? I will be checking my email into the evening to a certain point. I usually don't check beyond like 10:30, 11. But if you are studying and have a question, send me an email and I will try to answer it.